Our next speaker is Nicole Wanner. She is from the University of Minnesota and she's a third year PhD student. She will be discussing her research on the developmental cannabidiol exposure and how it increases anxiety and modifies genome-wide brain DNA methylation in adult female mice. Nicole?
Alrighty, thanks so much, Tia, and thank you everybody for tuning in today. Let me just share my screen. There, how is that?
Perfect.
Okay, great. So I will go ahead and get started. So cannabidiol, or CBD, is the primary non-psychoactive cannabinoid that's found in cannabis or hemp. And that's in contrast to the high-producing or psychoactive cannabinoid THC. And cannabidiol has appeared in the news quite a bit over the past few years, and there are a lot of anecdotal reports regarding its effects. But there's actually a mounting degree of evidence in the literature that it may be a beneficial adjunct treatment for some psychiatric conditions, including anxiety, depression, schizophrenia, and others. And there are a couple important gaps in the literature in this context. One is gene regulatory effects. There have been a number of different mechanisms posited, but that one has not received quite as much attention. And then additionally, to our knowledge, there are currently no studies examining the developmental consequences of exposure to CBD. And this is particularly important because developmental exposure to THC has been associated with detrimental behavioral outcomes from infancy all the way up through adolescence and young adulthood. So it's particularly important that we investigate CBD in this context as well. And the mechanism we're particularly interested in is DNA methylation. So as everyone here knows, DNA methylation can persistently impact gene transcription and therefore phenotype, and is also environmentally labile throughout the life course. And the resetting of DNA methylation marks during fertilization is a particularly critical window that's prone to disruption. And importantly, differential methylation has been identified in a number of different psychiatric phenotypes, including schizophrenia, autism, substance use disorder, and others. So our sort of main question for this set of experiments was to determine how developmental CBD exposure affects adult offspring behavior, and then also how it affects DNA methylation in several regions in the brain. So in order to answer this question, we wanted to evaluate genome-wide DNA methylation and also some behavioral outcomes in isogenic mice that were developmentally exposed to CBD. And to do this, we exposed nulliparous female mice to 20 mg/kg of CBD starting from 2 weeks prior to breeding and continuing all the way through when their pups were weaned. And then those weanling pups were then maintained up until adulthood, being abstinent from CBD. And then once they were 12 weeks of age, we then performed 2 behavioral measures, one of spatial memory and one of anxiety and compulsive behavior. And once those were completed, we then performed reduced representation bisulfite sequencing, or RRBS, which is a single nucleotide resolution method that assays about 3 million CpGs throughout the genome. And using this RRBS data, we then identified differentially methylated loci, or DMLs, and these were loci where methylation was significantly different between our CBD exposed and control F1 mice. And importantly, we enforced a minimum difference in methylation of at least 10% on these DMLs in order to try to increase the functional relevance of the results. And then after that, we gathered a list of all of the genes that contained at least one DML and enter that into a functional enrichment term program that would allow us to determine gene ontology terms and other categories that would help us understand the broad functional consequences of this exposure at the epigenetic level. So here I'm showing the results for our developmentally exposed F1 adults in the behavior assays that we performed. On the left, we have results for marble burying, which is a measure of anxiety and compulsive behavior. And on the right, we have Y-maze spontaneous alternation, which is a measure of spatial memory. And as you can see on the left-hand panel of both of these figures, CBD-exposed females, adult females that were exposed developmentally, had significantly increased anxiety behavior as adults, and additionally performed slightly better on the memory task in comparison to their control counterparts. So to our knowledge, this is the first measurement of developmental outcomes for exposure to CBD, adult outcomes for developmental exposure, excuse me. And the results appear to be sex specific, and additionally, this exposure is certainly not innocuous in the context of developmental exposure. Next, we wanted to evaluate our RRBS data examining genome-wide DNA methylation. And so here I have a histogram showing the count of differentially methylated loci, as well as the percent methylation difference for our two F1 brain regions, the cerebral cortex and the hippocampus, which are two regions that are broadly involved in psychiatric phenotypes. And so we found here that certainly developmental CBD did indeed result in shifts in the adult methylome in these two regions. There were approximately 2,200 DMLs in the F1 cortex and 4,200 DMLs in the F1 hippocampus. And in general, hypomethylation was more prominent, especially in the hippocampus, as we can see on the right. And there was an average shift in methylation percentage of around 50 to 60%. And I've also listed here some of the genes which contain the largest number of differentially methylated loci, so between 5 and about 10 DMLs. And I don't have time to go through them all individually, but they are notable in that they all have some degree of literature behind them for involvement in phenotypes like schizophrenia, depression, sociality, and autism, and others. So that was pretty fascinating to us. Additionally, I have also included the results of collecting all of those DML containing genes and placing those in a functional enrichment tool to identify some gene ontology terms and other terms that were most impacted by this exposure. And so you can see in the starred terms that I have here include neurogenesis and neuron differentiation, synapses, cell junctions, and the synaptic membrane, as well as some disease terms at the bottom, including neurodevelopmental disorders in general, as well as intellectual disability, epilepsy, which CBD is actually FDA approved for, and autistic disorders. And so these results were really exciting to us because they overlap quite a bit with the existing literature for direct exposure to CBD. It's been shown to increase neurogenesis in the hippocampal dentate gyrus. These results are for hippocampus specifically. It's been also been shown to be involved in synaptogenesis and signaling of a number of different neurotransmitters, dopamine, glutamate, serotonin, and others. And we, of course, can't make any sort of positive or negative claim regarding the effects on these disease phenotypes at this time, but certainly these results indicate that epigenetic mechanisms are likely to be involved in CBD's effects. And also, I didn't have room to display it here, but in the disease category at the bottom, these are only showing the top 5. But out of the top 20 terms that were most significantly enriched, 8 of them were for substance use disorder phenotypes. So that may be, despite not appearing in the top 5, that may be an additional phenotype that's particularly of interest for this exposure. So to conclude, we found that developmental exposure to CBD impacted anxiety and memory behavior, particularly in female adult mouse offspring. We also found that CBD shifted DNA methylation in both the cerebral cortex and hippocampus of those offspring. And functional enrichment revealed terms for neurogenesis, synapses, as well as several different psychiatric phenotypes. So there's lots to do in the future regarding this work. One would be to expand our behavior testing repertoire. Another would be to look at gene expression by RNA-seq, since we can't make any claims regarding the direction of effect for expression based on the DNA methylation alone. Additionally, it would be important to know whether these effects continue in subsequent generations through a transgenerational exposure. And additionally, we're interested in examining the effects of exposure during adolescence, as well as prenatal exposure with adolescent outcomes, since that is a common age of onset for a number of different psychiatric conditions. And lastly, I would just like to acknowledge my PhD committee, as well as the other members of the Falk Laboratory and our funding sources. And I would be very happy to take any questions. Thank you.
Wonderful. Thank you so much, Nikki. Before we move on to questions, I just want to put a plug in here really quick. Not next Friday, our session 4, which is on Friday, November 20th, will cover heritable impacts of tobacco, cannabis, and related products. And so if Nikki and Deidre's talks really piqued your interest, make sure to attend our next session, session 4 on November 20th. And so here is a question from someone in the audience. It is interesting talk. I am wondering how long and amount of dosage is needed to trigger F1 sex-specific phenotypes. Also, when during development.
As far as how long and amount of dosage, so the dosage of 20 mg per kg we based on the recommended dose for the FDA-approved version of cannabidiol for childhood epilepsy, which is called Epidiolex. And when you scale for body surface area differences between humans and mice, that actually ends up coming out to a more casual human dose equivalent of around 1.5 mg per kg. Additionally, our exposure window was fairly long, starting prior to breeding and going all the way to weaning. And that was in an attempt to mimic human patterns of dosing rather than try to isolate an exclusive window of acute exposure. But we do have some plans to expose exclusively during gestation in our next experiments to see if that has any influence on the results.
All right, wonderful. And then there's time for one more question. I'm just going to ask it really quick. In your next behavior test that you'll do in these future generations, what psychiatric disorders or certain psychiatric phenotypes are you looking for with your behavior tests?
There's the literature with CBD is pretty expansive as far as direct effects, particularly in animal models so far. There's been a more limited number of human studies compared to THC. But ones that I'm particularly interested in are anxiety and autism. There's some evidence for pro-social effects with direct CBD exposure, but whether or not that is more of a detrimental influence in the developmental context is something that's interesting to consider. So as well as the substance use phenotype as well, since that appears pretty prominently, that's also one that we plan to pursue.
Oh, that's exciting. Well, wonderful. Well, thank you so much, Nikki.